Hello fellow scientists, I'm Daniela.
And I'm Daryl via a phone call. COVID cases are still rising.
Today, we are conducting an amazing experiment entitled Biological Molecule Home Experiment. Biological molecules or biomolecules are chemical compounds that mostly the principal types of it can be found in living organisms. Biomolecules have varied in a wide range of sizes and structures, performing a vast array of functions, having four major types, such as carbohydrates, lipids, nucleic acids, and protein. These biological molecules are essential and are the fundamental building blocks of most living organisms, wherein the existence and significant concentrations of biomolecules are vital for cells to function properly.
On the other side, enzymes help to speed up chemical reactions in the human body as they bind to molecules and adapt in specific ways. Enzymes are essential for respiration, digestion, muscle and nerve function, and many arrays of roles they play essentially.
The objectives of the experiment are the following: to deepen knowledge on biological molecules and enzymes, to determine how enzymes speed up biochemical reactions, to determine how enzymes contribute to the digestion of food, and lastly, to determine what factors affect the enzymes to work properly.
The following questions are sought to be answered through this experiment. First, how do biological molecules and enzymes relate to each other? Second, how do enzymes speed up biochemical reactions? Third, how do enzymes contribute to the digestion of food? Fourth, how does temperature affect enzymes to work properly? And fifth, how do environmental pH conditions affect enzymes to work properly? In this experiment, the dependent variable is the specimens containing biological molecules, which are white bread loaf for carbohydrates, fried egg white for protein, and cheese for fats. On the other side, the independent variable is the enzyme bromelain found in the pineapple fruit juice. The alternative type of hypothesis was adapted in this experiment. There is a significant reaction between the biomolecules, carbohydrates, protein, and fats, and the enzyme bromelain. If the specimen containing biological molecules will be submerged on the enzyme bromelain, there will be a significant reaction to be observed. There are varying factors that affect the enzymes to work properly, such as temperature, pH meter, and material composition of the specimens. If the temperature is in normal body temperature, then the reaction would take place accordingly. If the enzymes are in a not too acidic and not too basic environment, then it would take it to function properly. If the material composition is light, then it may be digested or dissolved faster. Fiber content affects enzymes to work properly. If the fiber content of a certain specimen is low, then it may also be fast to dissolve, deform, or digest.
Now, let's start conducting the experiment. Here are the materials we used in this experiment: 3 containers of the same size, 20 grams of cheese, 20 grams of white loaf bread, 20 grams of fried egg white, pineapple fruit, blender, and knife. The skin of the fresh pineapple fruit must be removed. The pineapple must be blended and crushed until it becomes fairly smooth juice with a pulpy mixture. The 3 specimens containing biomolecules should be placed into the enzyme bromelain, which is present in the pineapple. Containers must be labeled with protein, carbohydrates, and fat to identify the specimen correctly. The specimens must be assured that it has been submerged into the pineapple juice for the accuracy of the results to be obtained. Specimens must be observed within an hour if significant changes have been seen. Observation must be recorded before, after 30 minutes, and after an hour.
With the results, we finally concluded that white bread loaf is easy to digest since it has a low fiber content with only 0.6 grams, making enzymes work on it rapidly. Fiber is a type of carbohydrate that the body has difficulty digesting. Though carbohydrates are converted into sugar molecules, fiber can be digested. Therefore, lower fiber content means a higher increase in digestion time. The rate of digestion of the enzyme bromelain present in the juice to the fried egg white is low. The egg white contains 0 gram fat or less fat and 0 gram fiber. Thus, it is way easier to digest. In this scenario, the fried egg white has notably seen less reaction with the digestion catalyst, but a difference in the juiciness of the juice was seen. On a negative note, there have been minimal observations on the reaction with only the participation of juice taking place in the reaction. One factor might contribute is the volume of the juice, which is way lesser than the higher volume of the egg being submerged. In this experiment, it has been seen that the cheese has deformed halfway. Hence, the bromelain enzyme somehow were able to catalyze the reaction, as there is also an increase in the volume of the juice, which can be said that cheese can turn into a liquid form once digested or melted. In this experiment, the temperature was logged at 30 degrees Celsius, which, hypothetically, may be a factor that the reaction is slow in progress. Also, a factor that may cause the enzyme not to work properly is the alkalinity and acidity of the environment. Enzymes are sensitive to the pH condition of the environment, whereas they don't work properly in such areas that are too acidic or too basic.
Thus, we made our hypothesis arrive to the manner it should be answered. This has been Daniela Manas,
And this has been Daryl Chirihinho.
scientist of present and future times. We wish you well and more reactions to come. Goodbye and good day, everyone. And cheers.